Hi, I'm Krishna Bera from Cognitive Science Lab, IIIT Hyderabad. Today I will be talking about motor chunking in internally guided sequencing. Motor chunking is a behavioral phenomena observed during sequence execution in many paradigms. It involves practice-induced consolidation of individual movement elements into clusters that allow efficient execution of the multi-element sequence. The benefit of such segmentation is that it results in an automatic control with reduced memory load and cognitive demand during sequence execution. Studies have shown chunking in a variety of tasks. In SRT and DSP tasks, the participants execute a sequence of key presses in response to a fixed set of visual stimuli. In the M-N task, the visual stimuli are presented as illuminated squares on a grid. The participants discover and learn the correct order of the sequence of key presses by trial and error. Such canonical paradigms involve externally specified sequencing, where the sequence of motor actions to be performed is contingent on externally specified visual stimuli and is not dependent on the participant's plan or volition. However, many real-life motor skills are internally guided tasks, where the sequence of motor actions is self-initiated by choice or some internal plan. Figure tracing is an example of externally guided motor task, whereas drawing is an internally guided motor task. In light of this differentiation, we investigate chunking in internally guided sequencing. We employ an adapted version of the grid sailing task first proposed by Fermin and colleagues. The task involves navigating a cursor from the start position to the goal position on the grid. The possible cursor movements and corresponding key press buttons are defined by a key map or KM. Each individual path or trajectory from start to goal position constitute a novel sequence of key presses. The repeated sequential execution of key presses during grid navigation amounts to sequence learning in an internally guided task. The repeated execution of these trajectories results in learning a self-generated, voluntary sequence of key presses. The figure shows key map defining the association between possible cursor movement directions and key press buttons. Here, the black cursor is shown initially at the start position marked as a green tile. The sub-goal is marked in red and the goal in blue. Participants first learn the key map by trial and error. Then, using the learned key map, they navigate towards the goal position via the sub-goal. Further practice allows them to optimize the trajectory and define the optimal or minimum steps trajectory. Participants score a maximum reward of 100 points on all optimal trajectories. An example optimal trajectory is shown here. The figure here shows a trial outline. Participants get 9 seconds to solve each trial. Each trial feedback consists of the number of moves in the traverse trajectory and the reward score earned in that particular trial. Our study hypothesizes the role of chunking in practice-driven performance improvements in GST. 15 participants performed GST on a 10x10 grid with single SG condition using the same key map. They performed 60 trials with the first optimal trajectory that they discovered. To examine the general learning in GST, we extracted successful trials where participants traversed an optimal trajectory and obtained a reward score of 100 points. Repeated measures ANOVA shows effect of trials on mean execution time, clearly indicating the learning in GST. The figure shows typical behavioral phenomena during discrete sequence production task. It shows how RTs vary across key presses in various phases: chunk initiation, execution, and concatenation. Based on a similar general profile of motor chunking, using Wilcoxon-signed rank tests between successive key press RTs, we employ 3 working rules to identify chunks. The initial element in a chunk is typically characterized by a significantly higher RT due to the time cost of loading the chunk in the motor buffer. Successive elements with a non-significant difference or decreasing key press RTs are appended to the current chunk, and a significant increase in key press RTs denote the beginning of a new chunk. We analyze the chunking behavior in GST during early and late phases. The early and late phase correspond to the first and last 10 trials in the task respectively. A 2x16 within subjects repeated measures ANOVA reveal that the key press RTs are different in both the phases. Early phase RTs are significantly higher than the late phase RTs. We also found that the RTs across key presses are significantly different. The figure shows emergence and reorganization of chunks in 4 representative subjects. The RTs in both early and late phase are plotted across key presses in red and blue respectively. The area between the two lines clearly indicate practice-driven performance improvements due to chunking. The brackets in red and blue on top denote chunks in early and late phase respectively. We observe that the chunks evolve with practice as the chunk boundaries change due to repeated concatenation and segmentation. Chunk features change with dynamically evolving temporal patterns of sequence execution. We found that the number of chunks across subjects significantly decreased from the early phase to the late phase. The chunk length significantly increased from 4.1 in the early phase to 5.3 in the late phase. With practice, smaller chunks coalesce to form bigger chunks as chunk representations consolidate with practice. Such a reorganization promotes automatic control of sequential movements, as the integrated sequence representations can be readily loaded into the motor buffer as one single unit for efficient execution with reduced cognitive demand. In conclusion, we show evidence for practice-driven performance improvements due to motor chunking and internally guided tasks. We found that the chunking is subject and sequence specific. In line with previous findings, we showed that chunks facilitate efficient behavior and these clusters consolidate into fewer larger chunks with practice. We show evidence for spontaneous chunking without pre-specified or externally guided structures while replicating the earlier results with a less constrained internally guided experimental paradigm. Future work can experimentally probe theoretical questions about hierarchical reorganization, how chunks are formed and integrated into a multi-level structure. We can also shuffle start, sub-goal, and goal positions to change the trajectories such that certain chunks are preserved whereas others are not preserved. This might help us validate if chunks are crucial for efficient performance. We can also examine whether chunks are independent of the effector hand used to perform the sequences. Thank you. Please feel free to join the poster presentation using the link given here.